Hello, good evening, everybody. This is Anup Zutshi from the US.
Yeah, good morning, Dr. Anup Joshi. I think for you it is morning.
It is absolutely. It's about almost getting to be 8:30 in the morning.
Yeah, my name is Dr. Murugan, Principal of College of Pharmaceutical Sciences, Dayanand Sagar University.
Nice to meet you, Dr. Murugan.
Yeah,
Thank you so much for your invite.
Yeah, it's our pleasure to.
Good evening. Good evening and a warm welcome to the second day of our international webinar series, which is organized by the Department of Pharmacology, College of Pharmaceutical Sciences, Dayanand Sagar University. Modern pharmaceutical industries have faced challenges to deliver safe and effective medicines because of toxicity and side effects of the discovered drugs. Recent advances in system pharmacology aims to address these challenges. Hence, the objective of this webinar is to provide the researchers, academicians, and students an overview of system pharmacology approaches to accelerate drug discovery and development. On behalf of the management, faculty, staff of Dayanand Sagar University, I extend a very warm welcome to the speaker of the day, Dr. Anup Sushi, and all the participants of the webinar. I now request our Dean, Dr. V. Murugan, Dean, College of Pharmaceutical Sciences, Dayanand Sagar University, to introduce the speaker of the day, Dr. Sushi, to the participants of the webinar.
Good evening to all participants from India, and good morning to our speaker, Dr. Anup Joshi. He has obtained PhD from Indo-Banaras University. Right now, he is a Director of Translation Quantitative Pharmacology at EMD Serona Laboratories, is a division of Merck from Germany. Since joining the organization in June 2015, Dr. Jutsi has built a high-performing team of scientists to support projects from early discovery to phase 2 proof of concept studies and beyond. The team supports projects in immunology, immuno-oncology, and oncology across research sites within Merck. Dr. Jutsi obtained a BS and MS in pharmacy from Banaras University, India, and a PhD in pharmaceutical sciences from the University of Florida. He has worked in contact science research for 9 years and in pharmaceutical research for 20 years. Dr. Jutsi is interested in understanding the mechanism of new chemical and biological entities as drugs and their impact on physiological system of healthy as well as diseased individuals. Dr. Jutsi's work involves studying large biotherapeutics molecules as well as small genobiotics molecules. His research leads directly to predicting first-in-human dose escalation strategies, dose stopping criticisms, predicted changes to biomarkers and disease endpoints. This helps to design the optimum clinical trial to establish proof of concept. A hearty welcome to you, sir.
Thank you.
On behalf of the management, faculty, staff, and students of Dayanand Sagar College of Pharmacy, as well as the participants from other colleges and other states, and our team of Deba, then Muhammad Imit, Sadak Ali, those are really involved for the webinar. I welcome all the participants to the webinar. Now I request the speaker, Dr. Anup Joshi, to take over the screen. Please welcome me, sir.
Thank you so much, Dr. Murugan. I appreciate. Can you guys hear me very well?
Yeah, sure.
Okay.
Please go ahead.
Right. So, Dr. Murugan, thank you very much for this wonderful introduction, and thank you for inviting me. I'd also like to extend a very great thanks to Dr. Geeta, who had reached out to me earlier to see if I was willing to speak to you. And I'm always very excited about these types of opportunities I get, particularly interacting with students. So I thank you all for this particular opportunity. So what I hope to do today, and I hope to achieve today, in fact, is to tell you something about systems pharmacology approaches and the reasons why we have now started adopting this type of an approach in our drug discovery and development setup. And I think many of you probably will understand that as I go through these slides, but just a very high level outline of what I'm going to present, of course, is a case for why I'm interested in talking about this subject. So why do we need to accelerate drug discovery and development? What are the problems in it? Why is that important right now? And then an introduction to the concepts of systems pharmacology. What does this term really mean? What are its definitions? What's the difference between a systems approach to pharmacology and regular pharmacology? And then I'm hoping to give you a little bit of an example, a very simple case study to outline the type of work we do and the approach of the systems pharmacology as we apply it to drugs actually in the development pipeline. And then I'm going to try to make a pitch towards India. As Dr. Murugan told you, I am an Indian. I have grown up and lived all of my life in India. So I always try to take opportunities where I can contribute to anything that's happening in India, particularly in the disciplines that I am generally comfortable with. So I'll make my pitch towards India, and that's what I call my soapbox opportunity. Now, I think this goes without saying. You probably have read more articles about this than probably I have. But the cost of developing a drug and bringing it to market is extremely expensive. And I think most of you realize that it literally now has spiraled to a range between about $1 billion to $2.5 billion per drug. You can see in the table below a whole bunch of pharmaceutical companies, many of the top ones, and look at the average cost of developing a drug in their own companies. And on an average, even if you don't take the high and the low ends, but a median value seems to be somewhere around $3 to $4 billion per drug. Now that is very, very unsustainable, right? Because every drug that you are going to make is not going to make you money to the extent that you're going to recover all of that work. So at the end of the day, despite the fact that we have a very magnanimous, very forward-looking approach to healthcare, the end of the day, every pharmaceutical company is a business. And many of them are indebted to their investors. They have to return the investment value to the investors. And so it operates as a business. Now, except the fact that there is a scientific rationale behind the business, and it's related to healthcare, but at the end of the day, it's still a business. So profits are important to these types of entities, right? So one of those challenges now makes it, you can see it'll make it very unsustainable to have costs that run up to, 8 or 9 of tens of billions of dollars per drug. And that's not going to be acceptable for research entities. Why is this happening? When I was at Pfizer, which is about 10 years ago, we had done a very retrospective analysis of all of our data. And you can see an arrow showing you all the processes that occur from preclinical. So this CAN means candidate alert notice. That means a drug has now come close to the clinic in terms of its development cycle. Then it'll get into first in man, which is a phase 1 study, phase 2, phase 3. And hopefully, if you're successful, you can launch that drug and it'll be, be sent as a new drug application to a regulatory agency like the FDA, etc. Now, this was Pfizer's, in those days, this was Pfizer's attempt at trying to rationalize how many how many compounds need to be in the pipeline for you to be able to get 4 products at the end of the day. And what happened was that the biggest bottleneck we found was between phase 2 and phase 3. And instead of having a 40% success rate, we were down to about 11%. That's what we realized was the bottleneck in this pathway. And actually, if you look back and see what happens between phase 2 and phase 3, basically, this is the time when the drug's mechanism of action is tested in man, or specifically in a diseased individual. And so when you have a failure, what it is basically telling you is that the drug's ability to affect the disease was not realized and the mechanism failed. And so therefore you have to trash the compound. That failure basically led us to think about what were the possible causes of all of this. And so in my next slide, well, one of the things we also investigated was, why is this happening? What's wrong with us? Why can't we do this much better? The first thing we noticed was that most of us wanted to try new targets so that you had new approaches to affecting disease or managing disease. And whenever you are working with a brand new target, the background knowledge about that mechanism is poorly understood. Obviously, that becomes highly risky now. But then what's the advantage of doing it? Well, the advantage is if you are successful, then you are the only person on the market having a product that works through that mechanism. And so you have tremendous exclusivity tremendous pricing advantage, tremendous patent protection, so that at the end of the day, you will start to say to yourself, oh, this is a great risk for me to take, because the return, if it works, is fantastic. The problem, of course, is the returns also become affected, as you saw from the previous slide. So one of the challenges we have is everybody wants to have an exclusive target to work on. And the risk associated with this is also very high. Obviously, then you have to be able to tolerate the failures too. That's what we realized, which was the first thing. The second was a recognition of the processes that we were doing wrong, essentially. So one of them was no understanding of the physiology of the disease and the factors affecting it. So many times we would look at things like, oh, we've got to develop a drug against diabetes. Oh, well, diabetes is simple, you see, because all we will do is measure the blood sugar and we'll know that that is elevated, and we'll dose, and then the blood sugar will come down to baseline, and then we are all happy about it. And nobody starts to think about resistance. Nobody starts to talk about insulin resistance. Nobody talks about glycogen dependency, et cetera, and all the other related mechanisms connected with your blood sugar hemodynamics. That problem causes a lack of understanding of these mechanisms, so that when you go into the clinic and you try it out, suddenly you don't get the response you're hoping for, and you are left scratching your head. What went wrong? What did I miss? And without realizing, what the system did to you was it basically changed a certain set of internal parameters. Your drug became useless. So this was what we thought of as understanding the true physiology of the disease. The second was lack of understanding of the drug itself now. Basically saying, what exactly does my drug do? What targets does it mitigate? What proteins does it upregulate? What proteins does it inhibit? That became a problem for us to understand too. The third point was poor translation from preclinical to clinic. That means we saw great outcomes in the preclinical space. So whenever we use certain animal models or certain cellular analyses, everything looked fantastic. But the moment you went into man, the variabilities associated with it, the differences associated with human subjects, this data was not able to translate in a 1:1 fashion. That confused us about what went wrong. Now remember, guys, one of the things that happens in the clinic, whenever you're testing a drug, you only have two opportunities to adjust anything. The first is dose. The second is dosing frequency. So all you can do in the clinic, because remember, unlike a rat or a mouse that you can cut open to and measure anything, in the clinic, obviously, you don't have that luxury. So if something doesn't work, the next best thing that the researcher thinks of is, oh, well, let's increase the dose. Oh, let's change the dosing frequency. Let's dose more frequently. And the whole idea is to basically see if you can push up the drug levels in the blood, so that if something happens, then you're hoping that, well, maybe it didn't happen at a lower dose, it'll happen at a higher dose. Now you recognize all of the toxicity that will come along with that if you start increasing dose, because now you are exposing higher concentrations of the drug to the patient, and you're going to see the accompanying toxicity of it too, and that could become limiting. So that becomes a huge problem, and this is what we call as poor translation. We sometimes use wrong models, right? Sometimes what happens in biology or in pharmacology is, oh, we will use this type of rat. And then it turns out that that rat model was actually not the correct model, because it doesn't have anything to do with human disease. But when you get the results from that model preclinically, everything looks fantastic. All the curves look beautiful. You've got fantastic coverage from the placebo and from the control group. But in translating that to humans, nothing works. So that is what I mean by wrong preclinical models used. And then finally, lack of understanding of the variability in the data. So preclinically, what happens is you invariably operate at relatively higher doses. And so you do not tend to see a lot of the variability in the data. But when you go into human subjects, there is tremendous variability associated with age, with gender, with body surface area, with creatinine clearance, so there are so many variables that you have no ability to control preclinically, but this will show up in the clinic. And so you have no idea of how to understand that variability. Suddenly, what you saw as a fantastic response preclinically no longer shows that in the clinic because the placebo and the control groups overlap with your treated groups. Now you are not able to differentiate efficacy, and there is no story to tell. You have to stop. So these were basically all of the reasons for the failures as we understood it. So let me go ahead and introduce to you now some ideas of what systems pharmacology means. I apologize ahead of time a little bit because this is going to sound like a class lecture. And I'm sure many of you as students probably wouldn't be very keen on listening to a lecture at 6 o'clock in the evening and are probably already exhausted with your day. But if you bear with me, I just wanted to highlight some things about what systems pharmacology means. So I want to go back to the basics right now, if you don't mind. Everybody knows the basic pillars of pharmacology. So this is the fundamental concepts of pharmacology as a subject, right? The first one is pharmacokinetics, which means the ability to know how to get the drug to the site of action. The second is the binding. And the third is the downstream change in the biology that will occur. That's the pharmacodynamics, right? So literally these three pillars are most important to understand the pharmacology of any drug, right? So what is pharmacokinetics? You all know this, probably have taken many classes in your study on pharmacokinetics, and you've studied non-compartmental methods, perhaps you've studied compartmental analyses. Some of you might have also ventured into these physiologically based pharmacokinetic models. And essentially all you're trying to do with this is to understand that once you dose the drug, how does the drug get to the site of action? And during that process of getting from the dosing site to where it needs to go, it obviously encounters a whole bunch of variables along the way. So there is an absorption component, there is a distribution component, there is going to be binding to plasma proteins, there is going to be distribution into red blood cells, there is going to be clearance from all of your clearing organs, there is going to be non-specific binding to tissues before the drug literally circulates and gets to your site of action, whatever that is. Whether it's a tumor tissue, whether it is a diseased organ, etc., wherever the drug needs to get. The study of that subject is the study of pharmacokinetics. So many of you probably remember from your class notes, stuff like words like clearance, volumes of distribution, fraction bioavailable, etc. I'm not going to go into the details about all of that. Perhaps that will be something your faculty would be able to handle for you. The second concept is once the drug has gotten to its site of action, you have designed the drug to bind to something, right? Because that's what you've structurally modified a chemical to, that all it is supposed to now do is to bind to a target and something will happen after you do the binding. This is a simple bimolecular reaction, right? So here is your drug plus a target, it will form a complex. Now this complex will have many ways of disposing itself, and I'll come to that in a slide later on. But essentially it's a classic equilibrium reaction that we study for the binding, right? Once the binding happens, then you come to the third stage, which is pharmacodynamics. Now, once the binding has happened, hopefully now something needs to change. Whether the blood pressure comes down, blood sugar goes down, protein expression levels change, a downstream cytokine is released, some other biomarker changes that you're trying to connect to your target. So all of these types of biological measurements are made. So this is essentially what we obviously call pharmacodynamics. The only difference is that you expect the pharmacodynamics to be time bound too. Excuse me. So you have to show that the change is happening as a function of time longitudinally. So this is essentially the first three basic concepts of pharmacology. Now let's and I'll send you guys these slides. You can study them at detail later on. But essentially, you will see that the terminology that you get from various studies of pharmacology, like IC50, EC50, etc., are things that you have known: the potency, the maximum effect, intrinsic activity. All of these terms are very similar to the ones we use on a day-to-day basis for our research. Okay, now what is systems pharmacology? So all this while, I talk to you about the properties of a drug. That means the pharmacokinetic properties of the drug, the binding property of the drug, what it hopes to elicit from the pharmacological system. But we all recognize that a drug essentially is going to act on a physiological system. This physiological system is your body. I hope you realize that you are unable to change this system. This system is fixed. If it is in a disturbed state, it will remain in a disturbed state till you treat it. So essentially, the physiological system cannot be interfered with until and unless you get a drug into the system. So you can manipulate the drug, you cannot manipulate the physiological system. It is what it is. Now, what is this physiological system? Whatever it is, you can see it is impacted by age, disease, tissue type, the type of species this is, the gender, whether there are other concomitant medicines that the patient is taking, that will impact your drug too. You also recognize the other physiological variabilities that impact your physiological system. For example, the baseline of a particular protein that you're interested in studying. Diurnal variability. For example, in many of our hormones, what happens? Certain times of the day, the hormone is upregulated. Certain other times of the day, it's down. Those hormonal changes affect hemodynamics. So the blood volumes keep changing depending on the time of the day. So it depends on when you take the dose, your pharmacokinetic properties might change because of something called the diurnal variability. Turnover of the target, right? Nothing in the body is static. It's constantly being synthesized and degraded. Fresh tissue, fresh enzymes, fresh proteins are constantly being generated. That's called turnover. So you need to know and understand what that system is doing. Target homeostasis, right? So a target is considered to be in a steady state throughout its lifetime. And finally, target-mediated drug disposition, where the target affects the pharmacokinetics of your drug. This typically happens for large protein molecules, and this is what is called as TMDD. I don't know whether you have heard this in your pharmacokinetics class, but if you are studying the pharmacokinetics of large molecules, at certain lower concentrations, you will find that the drug binds to the target and the target internalizes the drug, affecting the blood levels of the drug, right? So there are the pharmacokinetic curves have a very unique feature to them when such a process happens. Let me basically now just highlight to you some of the basic concepts of pharmacodynamics. The reason I bring this up is because this is the subtext of what quantitative systems pharmacology means. Practically all of you know a binding reaction, right? Drug plus a receptor will form a complex, drug-receptor complex. And you can create these curves for either the drug or the receptor or the drug-receptor complex. Now, many of you also know these nonlinear function called the Michaelis-Menten equation, right? And the difference is that it is the same reaction as a simple binding reaction, except that there is a clearance term to the drug, because the drug is going to get cleared from the body, and the complex degrades or changes in a first order process to form a product. That's the only difference. Otherwise, you can see the similarity between a simple binding equilibrium reaction and a Michaelis-Menten. And you can then extend this Michaelis-Menten reaction to what is called as a target-mediated drug disposition reaction, where the receptor, as I told you, is not constant. It is synthesized and it degrades. And once the drug binds, the drug complex can be internalized into the cell. So this could be a cell surface receptor sitting on the surface of a cell. Your drug comes along, binds to it, forms a complex. That complex is internalized, right? It starts to affect this concentration. While this is happening, this receptor is not sitting idle. It is getting synthesized and it is getting degraded. And so these curves start to acquire this very funny shape, this inverse S shape. This, whenever you see for large molecules, you know you are in a TMDD range for the pharmacokinetics of that drug. The second concept I wanted to introduce was the concept of steady state or homeostasis. Every system in the body, every receptor, every enzyme has to be at a homeostatic state. It may fluctuate a little bit, but it generally remains within a certain steady state value. And whenever you have homeostasis or steady state, that can be mathematically represented by a zero order synthesis rate and a first order degradation rate. Many of you probably have heard of these terms from your first year chemistry classes: zero order reactions, first order reactions, second order reactions. And I'm positive many of you went to sleep when those lectures were being given, thinking, why the hell am I supposed to be worried about reaction rates? But here is where they become very useful to you. See, so if you had paid attention, then you would have been able to figure this out, that this is a simple mathematical equation that establishes something at a steady state. So if you understand this, this is the second important aspect that you should know for applying systems pharmacology approaches. And the third is recognizing that all pharmacological reactions are capacity limited, right? So every biomolecular reaction rather, in the body is always a capacity limited process. Notice the metabolic rate and look at the structure of the equations for metabolic rate. Look at the structure of the equations for the transport rate. Look at the structure of the equations for protein binding equilibrium, receptor binding, pharmacological effect. Do you notice a trend? Do you see how each one of them has a setup where at the end of the day, you always have these S-shaped curves when you plot it on a log x-axis scale. And these S-shaped curves always will have a non-linear part in the beginning, then you have a linear part in the middle, and then it starts to plateau upon the higher concentrations. So as your concentrations increase, this curve behavior is very consistent, and it is consistent for all physiological and biological processes. They do not just keep going on and on till infinity. They will plateau at some point, okay? So what happens is if you realize and recognize that, you recognize the types of equations that will drive any pharmacological change. So now you understand the pharmacokinetics, you know the binding, you know the potential pharmacodynamic changes that can happen. As long as you understand those pharmacodynamic changes as equilibrium, Michaelis-Menten, TMDD, the concepts of homeostasis and steady state, and finally, that every biological process is capacity limited. Essentially, you now have all of the substrate that you have for you to be able to apply this to a systems pharmacology approach. Okay. Now, what are the limitations? The limitations are purely data. The limitations are purely connected with information. The mathematics is not wrong. The parameters that you need to support the mathematics becomes the problem. Why? Because like I just told you earlier, some of these targets are so new, so novel, that very little biology is known from them. So either we first establish and understand all of the biology so that we can study that well. Otherwise, what can happen is you will be left with uncertain values in your analyses. Now that will cause certain uncertainty in your outputs, but one can also sometimes mitigate or at least understand the impact of those changes by doing what we call a sensitivity analysis. That means you change the value plus 10-fold, minus 10-fold, see what happens to the outputs. If the outputs change very significantly, you realize that that value is very important for understanding its impact on the pharmacology. So these are the ways we try to minimize some of these lack of information. So the biology, as I told you, of the target is poorly understood. Preclinical species, as I explained earlier, are inadequate to understand the pharmacology, disease causes changes. You probably recognize that, if you are a diabetic and have been diagnosed early, the physiological and the disease states that you are in are very different from when you have been diagnosed much later. Similarly, if you have a tumor, and I hope that never happens to any of you, but if you have a tumor and are diagnosed early, obviously the tumor is easier and can be managed very effectively. But if you're diagnosed late, you know what the outcome is. You probably metastasize the tumor, and then it becomes difficult to control. So every time, all of these possible changes can impact your analyses too. Literature data, particularly in the early stages of a target analysis, are very discordant, very different. One laboratory will publish a certain value for turnover. Like 3 months later, some other lab is completely on the other side of the equation. They have got a brand new value, and you're saying, oh my God, this is the same protein turnover data. Why is it so different? But it's their methods, their techniques. You're not able to find out why their numbers came out differently than somebody else. So that becomes a challenge, right? And so whenever you are running these types of clinical trials, it becomes a huge challenge to control all of this variability. And I think I explained that to you in the earlier slide. Let me do a quick case study. I don't want to spend too much time on it. I don't remember now how much time I have. Oh my God, I'm already 30 minutes in. So I need to give you guys an opportunity to ask questions if you have any. So let me do a bispecific antibody as a case study. Now, I want to explain to you what a bispecific antibody is. One of the things that has happened in the pharmaceutical industry of late is the recognition that many of these diseases are so complex, particularly inflammatory diseases, right? For example, many of you probably have already read the literature now or are aware of the fact that even diabetes begins with an inflammation. Cancer begins with an inflammation, right? Atherosclerosis or cardiac disease begins with an inflammatory event. So all of these inflammatory events basically are multifactorial, very complex events. So one of the things researchers in the industry have realized, and of course even in academia, have realized that you cannot control these diseases by only hitting one target, a monovalent target. You have to attack the problem from more than one side. And so the idea is we can do it two ways, right? One thing is we can develop a combination of drugs. So you say, okay, I will have drug A, it will do the following things. I will also put in drug B, and that will do the corresponding things. And the combination of A and B now will be more beneficial to the disease outcome than A or B alone. So that's one way to do it. But of course, you realize in that case, you're literally having to develop two drugs, A and B. And so if either one of them fails along the way, then you have to now find the other partner all over again. Whereas the idea is that if we can develop an antibody drug, a large protein molecule, and have two targets attached to it. So it's on the same molecule designed in there such that the first target attacks pathway 1, the second target attacks pathway 2 as if it's another drug. And one molecule now is basically affecting both targets or two separate targets. This type of an approach is becoming very common in the pharmaceutical industry. Practically everybody is in the middle of trying to achieve some bispecific concepts for their portfolio of drugs, right? And obviously we in Merck are no different. We are also doing the same thing. We have literally maybe about 20 bispecific antibody projects that I'm dealing with currently. But essentially, that's the concept of a bispecific antibody. So in this particular case, I wanted to give you an example of a bispecific antibody against a T cell target. You probably know what a T cell is. T cell is a lymphocyte in the body that floats. It's generated in the bone marrow and it floats around in the body basically. And it is a tumor killing cell, the body's own internal immune system that will try to attack a tumor. Now, the other side of the antibody is supposed to bind to a tumor-specific antigen. So that the idea with this whole thing is that my antibody on the one side will go and bind to a tumor. So the tumor expresses certain proteins on its surface. So the tumor-specific antigen part will bind there. And the other part will look for a T cell and bring it close to the tumor, attracting the T cell to the tumor. That way the T cell will recognize, the hope of course is that the T cell will recognize the tumor and attack it. So you know, many of you probably might have heard that this kind of a concept, which is called immuno-oncology, that means you are trying to stimulate your own immune system to attack the tumor that it has failed to recognize. So the reason tumors exist is because your immune system suddenly doesn't recognize the tumor. And most of the reasons why it doesn't do that is because the tumor is expressing proteins or secreting cytokines or secreting certain dummy molecules that the T cells think, oh, looks like a healthy tissue, and they don't react to it, right? So one of the things we would want to do is wake up the T cells and say, hey, this is not a healthy tissue. This is a diseased tissue. You have to attack and destroy it. So this is the science of immuno-oncology, where you're trying to stimulate your immune system to do the job for you, as it should have in the first place. So that's what we call as an immunostimulatory molecule. Now look at this very complex diagram I have. I don't want to scare you with it, but I want to highlight three important concepts in it. The first one is that this has three separate compartments. You can see one is the plasma compartment. Now, what is the plasma compartment? This is the compartment where you're going to put the dose in. So you see this little green arrow. So it says, here's where the dose is going to come in. This triangle represents your antibody, which is bispecific. But of course, because it comes into the blood, doesn't mean it'll stay there forever. It has to get cleared. And so this is a clearance term for the antibody. Now, what will happen to this antibody? It seems that the T cell target is right there in the blood itself, in the plasma compartment. It can bind to it and form a complex, and that can get internalized. Now, you don't want this to happen by itself, but that's an unfortunate interaction that will happen because the T cells are part of the plasma compartment also. They are part of the tumor also, but they are part and hanging around in the plasma compartment too. Equally, they can hang around in the peripheral compartment. A peripheral compartment, for example, let's say this is the lymph nodes, right? Or the spleen. So these are the tissues in the body that hold all of your immune cells together. So the immune cells can float around in the lymph. They can be reservoirs in the spleen. They can be in the adrenal glands, et cetera, et cetera. So this is all compositely called a peripheral compartment. So your drug can distribute there too. And when it gets there, it can bind to targets in that compartment too. So you can see the reactions that I'm showing you, equilibrium reaction, to form corresponding complexes. It can then go into your tumor, which is where you hope for it to go. You can't control the other reactions, but you're hoping that a significant portion of your drug gets into the tumor. Here, it can bind to the first target, as we have designed it to do, form a complex. So this is a dimer now, right? Two separate units forming a dimer. And then the second molecule, this molecule will bind to the second target now, right? And form the trimer. So if this reaction continues forward, you will end up with these trimeric species. Similarly, the molecule can first bind to the second target first. There is no guarantee that it will only follow this sequence. It can instead bind to the other side first, then form that dimer. It can then bind to the first target and form the same trimeric complex that you saw over here. Notice now, as you are watching this set of reactions, a lot of the complexes that we are creating are very similar in their properties, right? So essentially, this complex here, we found here too, and we found it over here too. So the parameters that I will implement for this complex whenever it comes up will at first attempt be very similar in all of my compartments. Now, sometimes you might argue with me and say, well, Anup, how do you know that this complex that you're forming in the tumor is going to be the same type, exactly the same type in plasma? I don't know that, but at least it's a very good first approximation. Now, at the end of the day, if it turns out to be different and my scientific experiments reveal that, then obviously I will change the values over here. But as a first approximation, it's a good place to be at to say, oh, this complex is the same as this complex here, is the same as this complex here. I should parameterize these equations the same way. So once you do all of that, you now set up a set of parameters in this setup and can use this to guide your research efforts, which is what happened with our case. Right from the beginning, our company people had produced 4 molecules, and you can see them sequentially numbered 1, 2, 3, 4. And we were asked, can we guys decide which one is the best one to advance? Obviously, you're not going to take all 4 molecules through your development cycle. That becomes extremely expensive. So you're not going to say, oh, let's just basically carry all 4 of them along and figure out, which one is going to be the best at the end of the day. That's not possible. It's very, very, very expensive. So you have to make a choice, right? Now, how do you make a choice? So one of the things we said was, oh, here are these 4 molecules. Can we run it in the model that I showed you just to see target engagement, right? And so you can see in this table that I have, it looks quite busy, but notice that it has a certain sequence to it and a certain color pattern. So 1, 2, 3, 4 repeats itself. These are the same 4 molecules. The dark gray, right, is a pharmacokinetic half-life that we simulated for 7 days. That means every 7 days, the blood concentrations will drop by a half. The light gray is 3 days. So the dark blue is 7 days, light blue is 3 days. Now, simultaneously, you can see in the color sequence, the gray represents once a week dosing, and the blue represents once every 2 weeks dosing. Okay. So what's the advantage of knowing this? Well, if you already know right from the beginning of your project that this target and this type of an antibody that you have developed will not work for every 2 weeks dosing, you can start planning ahead of time that you're going to dose this once a week. Now, what would be the advantage of once a week versus every 2 weeks? Obviously, that the less frequently you have to dose a drug, the market value for that drug is higher. Why? Because compliance is relatively easier. Many of these doses have to be given by an intravenous injection. Now, intravenous injection means you cannot have the patient doing this at home, right? So he has to go to the doctor. Once you have to end up going to the doctor, guess what? You have to pay for that service. A nurse will put an infusion line into you so that she can dose you, or she or he can dose you, right? You have to pay for that service. You have to take the time out from your day to go and sit in a doctor's clinic. It might take you 1 hour to hang out there. It might take you 4 hours before your dose is finished. Who knows? So you wasted the better part of a day. Literally every week, 4 times a month, you will have to do it. Now, obviously, if you are a cancer patient and it's a difficult situation for you, you might tolerate it. But if I were to tell you, instead of doing it every week, you have to do it only twice a month, wouldn't you feel better about it? Of course you would, because you don't have to waste so much time at the doctor's clinic for doing it once every 2 weeks. And if you had to do it once every 3 weeks, all the more better. But here is where it becomes extremely challenging now. Notice that for these 4 molecules, remember I told you the gray, this 2 set of boxes is once a week. The blue is once every 2 weeks. Now the green represents that you can have a molecule that will not stimulate. So this value where it says below 10 nanomolar concentration means that you are not going to overstimulate the immune system. Now, one of the things that is very dangerous to do is you do not want the immune system to get overexcited or overstimulated. And the reason is because if you do that, sometimes it becomes runaway. It's literally a runaway set of biological reactions that you will not be able to control. The patient will crash out and probably die. And that is a big no-no in a clinical trial. You do not want your patients to have a very severe adverse reaction in a clinical trial, because it basically stops your clinical trial dead in its tracks. So what happens is, one of the things we do is make sure that there are certain safety criteria that we don't want to exceed. And in this case, we had decided a 10 nanomolar safety cutoff, okay? You will see that only a few opportunities for molecules 2 and 4 is a green. Every other scenario was a red, a no. And so we then were able to tell the project team, hey guys, molecule 2 and 4 are the best opportunities we've got. The 1 and 3 does not work. And definitely this is not going to be every 2 weeks dosing. We have to struggle with once a week dosing. Now here's where, guys, commercial will come into play, right? The commercial group in your company will say, oh, you guys are stupid. We cannot sell a drug that is going to be dosed every week because the competition is already doing it once every 2 weeks. What's the problem? I mean, this is something that we will not be able to compete with. So those are other pressures that we have to work with to figure out how to manage our expectations and our drugs, right? So this is, in this particular example, as you can see, once a week was the best chance we had. And the molecules 2 and 4 were the best molecules we could select. You can see, without lifting a test tube on a project, a simple modeling exercise like I showed you in the previous slide is able to tell you and guide you on which molecules to take forward. So all of these are simulations, in silico simulations, computer-based simulations, and they are so informative about decision-making in your drug pipeline, right? This is what I wanted to basically highlight with this particular example. So I will conclude and, hopefully give us some time to talk about things. These are very complex biological systems, as I just told you, and systems pharmacology and PK/PD modeling are powerful tools to try to unravel some of these complexities. Early deployment of these tools, as I just showed you an example, very, very early in the process, can reduce the costs and eliminate bad actors from continuing forward. Because as I told you, the cost of drug discovery and development increases as you move from preclinical to clinical. Clinical trials are extremely expensive, guys. Just as an example, right? Whenever you do first in human trials, particularly for phase 1, remember in phase 1, all you're doing is testing for pharmacokinetics and safety. You're not testing for efficacy. So in these trials, as a single dose, just a single dose to 6 patients, not patients, excuse me, 6 healthy volunteers and 2 for placebo, right? Just a simple 8 subject study. 1 dose, collecting blood samples and sending the blood samples to the laboratory for analysis of drug content will cost you $350,000 to $400,000, just 1 dose. So it becomes tremendously expensive to run these clinical trials, okay? So the earlier you can get rid of the bad actors, bad drugs, bad compounds, the better off you are. So you can obviously streamline, as I just mentioned, the laboratory objectives in your projects and make very informed go, no go decisions, right? Rather than saying, oh, let's go into the clinic and see what happens. Because you will find many times your scientists saying like, oh, Anoop, the animal is not a man. The animal studies are not human studies. At the end of the day, we will only know the best is when we do it in the human trials. And people take the jump into the clinic and keep their fingers crossed and hope for a miracle to happen. And sometimes that, more often in fact than not, it doesn't. And so that whole program gets wasted, right? And becomes very costly and expensive. And management, typically the big bosses in the company, are always going to question you and say, what the hell were you guys doing? How come you couldn't figure this out? So those are the things that we hope to eliminate in the future, have go, no go decisions that can be made effectively. We are also, I wanted to make a pitch for this. We are trying to understand the relationship between binding to a target and its final outcome in disease. This is a huge effort that everybody's trying to understand, is the disease biology, because the disease is unfortunately not a static thing. saying you have high blood pressure doesn't mean it's always the same thing, okay? So this is something we are already working on. Now here is my India pitch. I wanted to basically pitch this to academic labs and academic institutions such as yours. Because number one, in India, one of the things we have is we are pretty good in mathematics, okay? Many of us are very good and trained in mathematics. And even if we are not, we can learn it if we are taught very well with it. We have a good substrate of biology trained workforce, as you are probably evident from your own studies and your own teaching staff, how great your laboratories and teachers are. And so the knowledge base is there. We are very good in computers. Everybody knows the whole country achieved success past the 2000 time frame was because of our computer-based studies, right? Simple in vitro testing, cell-based analyses can be done easily in India. And look at what this is. You know, it's a very low investment with very, very high returns, because you can essentially do this type of work with essentially just a computer and an imagination of all of your biological processes, and then get very good returns on that particular investment. All right. So and that's why I make this plug. We should do this more and more in India, because I think it's very easy to achieve there. So with that, I will stop and thank you for your attention. If you have questions, I'd be glad to take them. If you want me to talk about some subject matter that is of additional interest, I can obviously, as you realize, I do a lot of talking. I can do that too. So Dr. Murugan?
Yeah, Dr. Anup, really, it's a nice presentation. Really, we enjoyed the mechanism of action. Generally, we used to see in the lab, but now theoretically, we have seen your mechanism of action. It was really well explained. But my question goes to you is, you did not touch any molecular modeling. Whether in USA, they are giving importance for the molecular modeling before what you call, they go for the pharmacological screening.
Yes, yes, absolutely. So that's a separate group that happens in our chemistry functions. I'm not a specialized person for that, but all of the molecular interactions, trying to get the structure of the target proteins, the various hydrogen bondings, the various interactions with the amino acids in that structural pocket. All of that type of modeling is done in our chemistry functions. And you're absolutely right. It is done before a chemical synthesis process begins. So they will do a lot of structural modeling, both in the large molecule space and the small molecule space. And based on that, they make decisions on what type of structural entities they need to build onto some framework of a molecule. Yeah.
One more question. See, suppose I have synthesized a molecule, but that molecule did not fit into any of the receptors. How do you know that the molecule and receptor doesn't go hand in hand?
So if you have an intent that you have designed a molecule to do something,
Yeah.
and if it doesn't do it, usually in the pharmaceutical business, we will basically not consider it. We'll just put it aside. But if you wanted to investigate what it can do in terms of binding to any target in the body, we will send it typically for what we call as a setup screen. And all that happens is that those guys, it's a contracted work. We send it to them, and they make a 10 micromolar solution of the drug that you've sent them, and they screen it against every potential protein target in the body. And then we'll come back with the result, and they will tell you, well, this binds to muscarinic receptor 1 with a potency of approximately so many nanomolars or so many micromolars. And then it also has an effect on a T cell receptor, CD137, and it does this to that receptor. So you can then try to see, based on that analysis, which physiological system is this thing active against. And if it has got some value there, you can investigate it for making it into a project and then advancing it as a drug. Sure.
Yeah. Thank you, Anup.
Sure.
Yeah, thank you very much.
There are a few questions posted in the chat box.
Okay.
Akshata will ask you a few questions.
Okay.
Thank you, sir, for your wonderful presentation on the system of pharmacology approaches to accelerate drug discovery and development. I now read out the queries from the audience. Here is the first question. How important the drug repurposing is?
What does that mean? What does that mean, drug repurposing? I think I know what it means, but I'd like to at least get it clear, or unless you don't know that either.
Okay,
Repurposing, I would think means whether there is a drug which was developed for a certain disease state or a certain receptor, and then you're trying to use it for something else, correct? Isn't that what probably it means? I think that's probably, yeah, I probably believe that that's what it means. So yes, that happens a lot. You will find, for example, sometimes, and this happened when I was at Pfizer one time, it's not usually very common, but what we do is we have a project sometimes and we want to find other analogs. And so we will do a screen of a library of compounds that are already existing. And we suddenly find, oh, you know, this particular drug works in this particular indication also. And so can we try it out for an alternate indication? So yes, that happens. In fact, as you guys know, with the COVID situation, that's essentially how everybody tried to repurpose, hydroxychloroquine to the COVID situation, and then and of course tried everything with the COVID virus because nothing seemed to be otherwise working. So yeah, so that repurposing can happen. Depends on what the needs of the project are. It's a little expensive to do sometimes because if you have to select it from a whole library of compounds, it takes a while.
sir, there is another question. How an analyst or researchers get an idea about a particular old out marketed drug to repurpose?
Invariably for us, it might come through the information through a literature source. Sometimes what can happen is that when, as Dr. Murugan asked earlier on about modeling drugs in a chemical space, we start to notice patterns, right? So if you start to notice a pattern, you say, hey, you know, I think I've seen this in a similar molecule that was used for some cardiovascular disease. This resembles that molecule. Then you get prompted to see, let's see if we can recognize what this is. And when you compare it, then you say, oh, this is essentially this type of molecule. Let's see whether it works for our indication too. Now, of course, if it is already existing out there and it is already marketed and it is probably already expired on its patent, for us, there is no business reasons for us to continue with that molecule because it's not going to be giving us any advantage. So what we will end up probably doing is taking that molecule, recognizing its structure, modifying it, and registering it as a different molecule, if we wanted to pursue that, right? But as you know, the FDA also doesn't like that anymore. It has punished companies like the American Merck, and GlaxoSmithKline was punished a couple of times for doing these types of manipulations. So, but if you were to, based on the question you asked, whether we can and how we repurpose a molecule, that's essentially how we would do it.
Thank you for the clarification, sir. There is a next question.
Mm-hmm.
How important is in silico modeling in drug development?
So as you saw in my example, this was all in silico, right? We had not even done the first experiment yet. This was just a project in its concept stage. The project team wanted to use a T-cell engager and also a T-cell, a tumor cell binder as a bispecific. So everything that I just showed you was all in silico. So we have now, so I must admit that this is relatively new, okay? This whole systems approach, because if you were to go back about 15 years, this did not exist. It was very poorly defined. Now, because of all the problems and the expensive nature of drug development and the reasons for why we keep failing, this is something that we feel is important. So a lot of these in silico approaches have become extremely valuable. One which Dr. Murugan pointed out in selecting a chemical to process forward for the target. With the types of analyses I'm showing you, which basically incorporates all of the pharmacology also, drug behavior also, trying to understand what's the best scenarios that you have in your advantage and favor. So these approaches have become very, very common now in the pharma industry, and you will have specific business units devoted to doing this type of work now.
Thank you, sir. And there is a next question from the audience. How does system pharmacology influence drug targeting in molecular level using nanomedicines?
Oh, that's a very complex question. Nanomedicines. So nanomedicines have their own issues. You can get some targeting with that. The problem with these approaches is that the body reacts quite adversely to them. When I say adversely, I mean that the body is designed not to have particulate matter floating around in its system. So every time you have a nanoparticle or a liposomal particle or even a modified erythrocyte as a carrier system, what happens is that the reticuloendothelial system in your body, which comprises of your lungs, liver mostly, spleen, these systems will clear the drug away very quickly. So one of the biggest challenges for this type of therapeutic approach is that you have to be able to keep high concentrations or relatively high concentrations of your nanobodies in the blood for it to get to the target before the liver and the spleen and such of these clearing organs remove it from the circulation. So that's always been a huge challenge for this modality. But yeah, it's an emerging science. We do not have a lot of products with it yet, but hopefully in the future, we will see opportunities for developing it. Yeah.
Thank you, sir. Next question from the audience. Can specific immunostimulator be used for cancer therapy? And how sure will it target only the cancer cells?
You can't guarantee that it will only target the cancer cells. The hope is, though, that because of the way you have designed the molecular structure, that it preferentially binds to the tumor cells, okay? It is not going to completely guarantee that, but it preferentially binds to the tumor cells. So at least you've got some focusing ability by designing the molecule that way. Now, there are many immunostimulatory drugs that you probably have heard of. Even my company has a molecule called Bavencio, which is avelumab, a protein that binds to the PD-L1, the PD-L1 ligand, and that stimulates the immune system to attack the tumor. There is a whole bunch of drugs. You might have heard a name called pembrolizumab. I don't know the brand names, guys, because those are very specific to India. But pembrolizumab is the generic name for that drug. Nivolumab. These drugs essentially attack they stimulate the immune system to basically try to attack the tumor on its own. One of the things we have noticed, of course, is that if you can stimulate the immune system adequately, it does a much better job in killing the tumor cells than any of your drugs can do. Okay, your regular drugs are so much more toxic that you see a lot of side effects before you see any beneficial effects on the tumor. But these approaches are relatively easier to manage in the clinic. Of course, you see side effects, but they're relatively easier to manage. And the benefits that you get out of it sometimes are quite remarkable. Okay, very groundbreaking benefits in terms of changes to the tumor dynamics.
Thank you, sir. Next question from the audience. Like bispecific antigens, are there any polyspecific antigens which acts on many targets?
No, we haven't gotten to that yet. So right now we are still struggling at the bispecific level. And suffice to say, even this is quite complex. And we don't understand everything in it even today. So polyspecific or multiple targets on one molecule is a futuristic thinking. But we are probably not going to be there even in the next 10, 12 years. It'll take us some time.
Thank you, sir. And there is a last question. How does system pharmacology approach and marketing interact?
I mean, you are from one end of the discussion, one end of the spectrum of study to the other end. Systems pharmacology approaches essentially allow you to develop a drug with hopefully with less interruptions, less problems along the process of developing the drug. Marketing in of itself is a separate effort. I mean, it's not going to, marketing will only depend upon how successfully you have made the molecule. If the drug is very unique, it's obviously easier to sell. If the drug has not only unique properties, but is also quite safe in the sense that the side effect profile is very easy and manageable, then obviously it will sell very well and gets prescribed more often. But otherwise, these are kind of unrelated. Systems pharmacology is more a scientific approach to discovering and developing drugs, whereas marketing, of course, as you know, is just trying to sell the drug. That has to be based upon many, many other factors. Selling a drug in Japan, for example, has different criteria. Selling a drug in Europe has different criteria, and so on and so forth. And it also depends on pricing, right? Because it depends on what every government is willing to allow you to price the drug in their country. So all of those factors play a role. So unrelated to systems pharmacology, actually.
Thank you, sir. And there is one more last question from the chat box. System pharmacology and genome medicines. What is the future for this?
Very, very good, very high. Genomic understanding of your targets and how your drug changes the message or the RNA levels of your proteins is extremely important and a hot topic nowadays. Obviously, we would prefer to have protein levels rather than message levels, because RNA upregulation, what happens unfortunately is doesn't necessarily translate to protein. So sometimes it can get misleading. You might see a huge signal in your RNA sequencing data, but it doesn't necessarily translate to a biological effect. So those are confounding factors for that. But right now, that's the best we have. And that is a huge, huge topic of interest in our drug discovery systems. And obviously, systems pharmacology, which is essentially a mathematical approach to deconstructing your pharmacology, allows you to utilize those data from the genomic sequencing data, etc., to study the system better, of course.
Thank you, sir, for your clarifications. Now the session comes to an end. For vote of thanks, I hand over the session to Geeta ma'am. Thank you, sir.
Thank you, sir, for this wonderful presentation on system pharmacology. You made the topic very simple to the participants. On behalf of the management, staff, and students of DSU, I express my heartfelt thanks and appreciation to our speaker, Dr. Anup Suchi, for accepting our invitation to this webinar and sharing his valuable thoughts and findings in this important area of research in pharmacology. A sincere thanks to all the participants of this webinar for making this event a success. I would like to thank Dr. Murugan, Dean, for his support, vision, and commitment in organizing this event. I acknowledge the unwavering support in the conduct of this webinar from our promotion team, Mrs. Deepa, Mr. Shaukat Ali, and Mr. Hemant and all our faculty members. Thanks to one and all. And with these warm words, we move to the end of today's webinar. I request the participants to fill the feedback form and submit. An e-certificate will be sent to you soon. Thanks a lot. Thank you.
But I just wanted to also thank all of you for spending an evening. I know it's late in the evening for you there. For spending the evening with me. I appreciate it, and I definitely appreciate the opportunity. I love to do these types of talks. So good luck to you all. If you have any questions of me, feel free to ask. I will send the details to your professors, and if you are interested, you can connect with them. So thank you again, guys. Have fun. Very, very good evening to all of you.
Good evening. Thank you, Anna.
Good morning
Bye-bye.
to you.
Yeah. Thank you.
Bye-bye.
Bye. Bye.